السلام عليكم ورحمة الله وبركاته. محاضرة اليوم هتبقى على lecture 12 and 13 على Antimicrobial Chemotherapy. We are going to discuss important topic. What are the antimicrobial agents which affect microorganisms for treatment of infectious diseases. We are going to give some important definitions for the antibiotics, the chemotherapy, for selective toxicity. Describe the difference between bactericidal and bacteriostatic, and what is the narrow and broad spectrum antibiotic. Describe in detail the mechanism of action of different antimicrobial agents, and the part 2 of the lecture will be dealing with the different mechanisms of antimicrobial are going to discuss the action, how the antimicrobes act or affect the microorganism, destroy the microorganism, and also in reverse we are going to describe what are the different antimicrobial resistant mechanisms. Also the side effects of these antibiotics or antimicrobial agents. The elements of anti-chemotherapy involve the microorganisms and the drugs and the host. In our lecture, these two lectures will be dealing with interaction between the microorganisms or microbes and the drug. How the drug will affect the microorganism, is it effective of destroying these microbes or not, and how far its effectiveness, how to measure this selective sensitivity. Also in reverse, how the microbe will produce different resistant mechanisms to resist this drug action. An antibiotics defined is an antibiotic is defined as a product produced by living microorganism to suppress or inhibit the growth of another living microorganism. So it is a product of a living microorganism to inhibit another living microorganism. But however, like the penicillin, penicillin is actually a product of penicillium fungus, and as we see here, this is the staphylococcus, and it produces penicillin, which inhibits the growth of Staphylococcus aureus around the growth of penicillium. However, some of them are semi-synthetic or synthetic. And the term of antimicrobial is usually is preferable. Antimicrobial drugs are usually effective for treatment of infectious disease because of their selective toxicity. What is selective toxicity? It is the ability of the drug to inhibit or kill the growth of an invading microorganism without harming the cells of the host or of the human being. And this selective toxicity usually comes from structural or biochemical differences between the microorganisms, microbial cells, and the human or host cell. This is the cause of selective toxicity. However, this selective toxicity is not absolute. Sometimes it is relative. For example, for that selective toxicity, we discussed before the microbial structure. We know that the bacteria have a cell wall and then have a cell membrane, a cell wall and a cell membrane, and this cell wall is characterized in the structure of bacterial cell. It is absent in human cell. So if you use drugs which inhibit the growth or suppress the growth or the formation of the microbial cell wall, it will not harm the human being. Antibiotics are usually classified according to the mechanism of action. This is a major topic of or major core of our lecture today, or either to bactericidal antibiotics or bacteriostatic, or according to their spectrum. Bactericidal, from its name, this is an antibiotic which will kill all the microbial population. Bacteriostatic, it will inhibit the growth or stop the growth of these antimicrobial agents. Broad spectrum antibiotic, they will inhibit, it has a broad range of microorganisms, can suppress or can affect this microbe. It can affect both gram-positive, gram-negative, maybe tuberculosis, TB bacilli as well, and so on. If it is a narrow spectrum, these antibiotics or drugs will only affect the gram-positive or the gram-negative bacteria. So according to the mode of action, the antibiotic which kills the bacteria called bactericidal, which stop or inhibit the growth of bacteria called bacteriostatic. According to the mechanism of action, according to the microbial bacterial cell structure, we have the specific from the outside, we have the cell wall, we have the cell membrane. It may act on the protein sense, inhibition of protein senses. It may act by inhibiting the nucleic acid senses, or maybe a competitive inhibitor for an important metabolite for the microorganism. Inhibition of the cell wall senses. We have beta-lactam and non-beta-lactam antibiotics which act on the selective toxicity here, because the peptidoglycan layer present in the cell wall, this is site of action of the antimicrobial agents, where inhibition of the peptidoglycan senses or elongation, this is the target of action of these drugs. So stopping or affecting this target will lead to defective senses of the cell wall and lead later on to lysis of the bacterial cells. Affecting the cell membrane and especially the polymyxins or polyenes or amphotericin B, but these drugs are highly toxic drugs. Affecting the protein senses in bacteria, and as we know, there is structural differences in the ribosomes, whereas there are 70S ribosomes in bacteria and 80S ribosomes in human cells. So here comes the selective toxicity, where these drugs are selectively affecting the bacterial senses of the drugs proteins, affecting inhibition of the nucleic acid senses, either inhibition of RNA polymerase or like the rifampin, or inhibition of DNA gyrase, DNA replication, inhibition of the helicases or topoisomerases. This is action of quinolone antibiotics. The fifth group of mechanism of action is the competitive inhibitor for an essential metabolite, where folate in the sense of folate senses, interruption of an essential metabolite for the bacteria, this will lead to the defective the inhibition of the bacterial affect the bacterial cells and the mechanism, and a fifth mechanism of action of antimicrobial agents. Another figure, we have the cell wall inhibitors block the synthesis and repair of the peptidoglycan layer, which is selective material structure present in the microbial cells. So this is the target of action. This is a cellular target. We have the beta-lactam group and non-beta-lactam antimicrobial agents. The cell where the cell membrane affecting the cell membrane structure causes loss of selective permeability, like the polymyxins, like the amphotericin B, affecting the DNA senses, like the quinolone antibiotics, characterized by affecting the DNA senses, DNA helicases and topoisomerases, or inhibiting RNA polymerase, like the rifampin, affecting the protein senses. And as we said, there is different structural differences between the ribosomal subunits in the bacteria, where it is 50S and 30S, in contrast to 60S and 40S in human cells. So this means the selective toxicity of these antimicrobial agents, or maybe acting as a metabolic inhibitor. It suppresses the interfere with an essential metabolite for the bacteria, leading to bacteriostatic or inhibit the growth of the bacteria. First mechanism of action, inhibition of cell wall synthesis. It is, as we said, the bacteria have a unique bacterial cell wall structure and function, so it is the site of selective toxicity. Penicillin, cephalosporin, vancomycin will interfere with cell wall synthesis, will finally interfere with the elongation and transpeptidation and formation of the peptidoglycan layer, and this interruption of the peptidoglycan synthesis and eventually lead to cell wall lysis. Beta-lactam will inhibit the final step of the synthesis of peptidoglycan. However, vancomycin inhibits the early steps for the for the synthesis of peptidoglycan layer. That's why vancomycin is a preferred or the primary drug used of beta-lactam resistant Staphylococcus infection, the methicillin resistant Staphylococcus aureus, which is a very common microorganism to be isolated from hospital acquired infections, especially in the intensive care units. Penicillin selectively interferes with synthesis of bacterial cell wall. As we said, its structure is not found in mammalian cell. Selective structure present only in bacteria, and that's why penicillins are inactive against organisms devoid of a cell wall. As we take in the microbial cell wall structure, we have the some bacteria are devoid of or cell wall negative or devoid of cell wall, like the mycoplasma, like the protozoa, the fungi, the viruses. So these are penicillins will not be effective. Actually, penicillins also should act on rapidly active replicating drugs. Glycopeptides like vancomycin, as we said, it interfere with binding of D-alanine amino acids, inhibit the addition of subunits to the backbone peptidoglycan synthesis, and they are actually very effective against the MRSA, methicillin-resistant Staphylococcus aureus. Like penicillin, cephalosporin, it inhibits the beta-lactam final steps synthesis of peptidoglycan by binding penicillin binder protein in the cell wall and prevent transpeptidation. Virtually all antibiotics will inhibit bacteria cell wall synthesis are bactericidal, because after the interfere with the cell wall is an integral part of the microbial cell, and eventually it will lead to cell lysis, osmotic lysis of the cells. Second point for interfering with the mechanism of action of antimicrobial agents is the inhibition of the bacterial cytoplasmic membrane function, where some antibiotics will lead to disruption of cytoplasmic membrane and leakage of the cytoplasmic contents, leading to bacterial cell death. However, selectivity is based here on the differences in types of lipids in microbial cell membrane, and there is usually a narrow spectrum or narrow margin of selective toxicity. Examples for the antimicrobials affecting the bacterial or microbial cell wall is the polyenes like the amphotericin B. Third important mechanism of action is the inhibition of bacterial or microbial nucleic acid synthesis, inhibition of nucleotides, inhibition of replication, or stop the transcription, which act on any step of DNA or RNA replication. Rifampin will block the RNA polymerase, so it affects RNA transcription. Quinolone, it affects the DNA helicases or topoisomerases, which are or DNA gyrases, which are very essential for the microbial replication of bacterial DNA. interfering by fluoroquinolones are uniquely inhibit the replication of bacterial DNA by interfering with action of DNA gyrase during the bacterial growth. This is leading to stopping the inhibit the rejoining step and thus causes cell lysis by inducing cleavage in the DNA. This is an important mechanism of action, and usually quinolones are used in urinary tract infection mostly. Protein synthesis, as we said, there is a difference, important difference between microbial ribosomes and the host or human ribosomes. This differences in the ribosome subunits makes the antibiotic selectively affecting the bacterial cells and little harm in human cells or and so on. So antimicrobial have a selective action against prokaryotes because the bacteria is prokaryotes. As we said, this is a major selective toxicity comes from here, from structural difference. Aminoglycosides affect the 30S subunits, leading to misreading of messenger RNA. It is a broad spectrum used against gram-positive, negative, and some of the gram-positive bacteria as well. There are different aminoglycosides. This is a large group of antibiotics. It can involve streptomycin and gentamicin and other groups of antibiotics. Last step of the mechanisms of action of antimicrobial agents is the competitive inhibition. Competitive inhibition, this is which is a chemotherapeutic agents will compete with an essential metabolite for the same enzyme. The para-aminobenzoic acid is an essential, if we come to that figure, is an essential metabolite for folic acid synthesis in bacteria. So which is essential for later on for nucleic acid synthesis. Sulfonamides are structural analogues for para-aminobenzoic acid, and they will compete with the enzyme, thus inhibiting the folic acid synthesis. So there is a competitive inhibitor. Para-aminobenzoic acid is essential metabolite to synthesize the folic acid, and sulfonamide is a structure for that para-aminobenzoic acid, so it will inhibit the formation of folic acid, which is very essential for the synthesis of nucleic acids in the bacteria, so interfering with the DNA replication later on. So a combination known as co-trimixazole shows greater antimicrobial activity than equivalent quantities of either drug alone. So it is a combination, co-trimixazole, sulfonamides. Synergistic antimicrobial action of these two drugs together results from two sequential steps. The synthesis of tetrahydrofolic acid, as we look here, we have the para-aminobenzoic acid, betridine synthase enzyme will convert microbial betridine synthesis to dihydrobutyric acid, and this will be converted to dihydrofolic acid and then tetrahydrofolic acid. What happens? Sulfonamide is a structural analog to para-aminobenzoic acid. So this is a competitive inhibitor with the para-aminobenzoic acid structure analog together. So it will suppress this enzyme and will not affect the progress of formation of folic acid, which is essential to form the nucleic acid nitrogenous purines, which are very important in synthesis of folic acid. So this is a role of sulfonamides. Trimethoprim will affect the last step for formation of folic acid. So there is to repeat, it is a chemotherapeutic agent, competes with an essential metabolite for the same enzyme. Para-aminobenzoic acid is a very essential metabolite for the organism for formation of folic acid, as it is a precursor of folic acid. Sulfonamide is analog to para-aminobenzoic acid, so they enter into the reaction in place of para-aminobenzoic acid and compete with the active center of the enzyme, so it will inhibit the tetrahydrofolate formation, which is essential for forming in the nitrogenous basis of the DNA. So to summarize, this is a primary lecture. We have different mechanism, different terminology. We have different mechanisms of action. The word antibiotic is a term for a drug which is used for produced by living microorganism to suppress the growth of another living microorganism, like the penicillin. We have different mechanisms of action of antimicrobial agents depend on the different structural differences between the human cells as well as the microbial cells. And these differences are the basis for the different mechanisms of action. So we have some terminology. Chemotherapeutic, which is a chemical substance used for treatment or relief or prophylaxis of a disease. The word prophylaxis, the use of drug prophylaxis to prevent imminent infection of a person at risk. Antimicrobial chemotherapy, the use of chemotherapeutic drug to control infection. Antimicrobial, so to control infection. Antimicrobials, it includes inclusive term for any antimicrobial drug regardless of its origin. Antimicrobial could be antiviral, could be antibacterial, could be antifungal, could be antiparasitic. So antimicrobial, it is a terminology affecting any drug which can affect any microbe and microorganism regardless of its origin. Antibiotic is substance produced by a natural metabolic process of some microorganism which inhibits or destroy the growth of another microorganism. It is a substance produced by an living microorganism to suppress or inhibit the growth of another living microorganism. Semi-synthetic, which are chemically modified in the lab after being isolated from the natural. So we take this antibiotic and we make some modifications in the lab to become semi-synthetic. Synthetics, it's purely produced in the lab. Chemical reactions to produce the drug in the lab. We have broad spectrum. As we said, these drugs which affect the broad spectrum, they work and they suppress the antimicrobial, the gram-positive as well as gram-negative. We have the narrow spectrum, which are these are the drugs which affect the either gram-positive, gram-negative. Some drugs are anti-tuberculous only, used for treatment of TB, some drugs, and so on. This is a summary. You will take it in more detail in pharmacology, according to the dividing the groups of antibiotics